The bitter, sweet, and savory taste sensing pathways video, made by Cassidy Czombel. This is Frank. Frank's favorite thing in the whole world is food. But one day, Frank got to thinking. How can someone really tell the taste of all these different foods apart? Upon taking a closer look, each of these tastes have their very own processes to signal to Frank that he is tasting sweet, bitter, or savory items. In order to taste the sweetness of ice cream, the savoriness of meat, and the bitterness of lemons, G protein couple receptor pathways are required. Let's first discuss what a G protein coupled receptor pathway is. A G protein coupled receptor complex is located in the plasma membrane of the taste receptor cell. The complex consists of receptors and an alpha and beta gamma component. When the taste molecule comes in, it binds to the G protein coupled receptor, which disassociates the alpha and beta gamma subunits from each other. The alpha subunit then connects to the phospholipase beta-2 complex, which activates the second messengers IP3 and DAG. From here, IP3 travels to the endoplasmic reticulum and activates calcium ion channels. This activation releases calcium ions from the ER and into the cell. Calcium is crucial to these pathways because calcium is used to activate TRP channels, which allows the flows of sodium ions into the cell, leading to depolarization. The taste of bitter, sweet, and savory items all use this pathway. So what is the difference? It is important to look at the beginning of the pathways to answer this question. The sweet sensing pathway consists of a dimer receptor with receptors T1R2 and T1R3. These two receptors are crucial for sweet sensing pathway because only when both of these receptors are present and functioning will the sweet taste be accepted. The umami or savory sensing pathway is almost identical to the sweet sensing pathway, except the umami pathway uses the protein receptors T1R1 and T1R3. The bitter pathway is different from both the sweet and umami sensing pathways in that it uses a completely different class of receptors. In the case of the bitter sensing pathways, monomer receptors classified as T2Rs are used instead of T1Rs as in the sweet and umami pathways. Thus, we have the ability to distinguish between many different tastes from many different foods. While our taste sensing processes are not limited to these, three pathways are the sweet, bitter, and savory pathways. All of these tastes are sensed by G protein coupled receptor pathways, and the main difference between each of them are the receptors present at the beginning of the pathway. Now Frank doesn't have to worry that his ice cream will taste like bitter or his turkey dinner will taste sweet.